Hi guys, this is Miss Hart-Townsley, and I've had an incredible amount of questions over this 1.05 Earth's early atmosphere assignment. And so what I would like to do is just kind of take you through. The first thing I want to point out is right here at the very bottom of my first page, it gives me my objectives. And so as we can see, the lab is going to deal with this last objective, which is explaining the connection between the beginning of life and the universal genetic code of almost all organisms. All right, so when you email and you say you do not understand the lab, let's make sure that we've looked through the module and let's covered all the information. And so as I click through this lab, you should be taking notes over everything that you see, including the interactives. Okay, so you notice how I have 4 pages of information. Okay, and in those 4 pages of information, we talk about the different hypotheses of the beginning of life, RNA world, and whatnot. We also have a really good summary at the end of the lab, which is called the Let's Review or Science Review. And so make sure you've also done that. On that 5th page, after you've taken notes all over the first 4 pages, you can see once again we have the summary, we have a reminder of the objectives, and then you have the assignment. Okay, you guys are talking about confusion about the chart. Well, let's look and see what's going on. So it says gray whales and yeast cells do not seem like they have much in common, but their DNA molecules use the same coding rules to build protein and direct cell function. This universal genetic code that is shared by all living organisms gives possible support for many of the hypotheses about the origin of life. In this assignment, you will examine genes of several different species and apply this information to the hypotheses discussed in the lesson. All right, so the letters that you're going to be looking at represent the amino acid sequence, the nucleotide sequence of those DNA, of those organisms' DNA. Okay, so I'm going to open up the assignment, which tells me a little bit of background information about the genes that I'm using. Okay, so we're going to be looking at the cytochrome C gene. This is a metabolic protein that helps release energy from food. Scientists study similarities such as the gene that codes for cytochrome C in hopes to learn more about the universal genetic code and the relationship between living organisms on Earth. All right, the charts below list a portion of the amino acids found in this protein of several different organisms. Use the chart to compare the amino acid sequence in humans to the sequences of the other organisms listed. For each organism, identify any amino acid that is different or missing when compared to the amino acids in the human sequence. Okay, so we're going to compare all those different animals to the human chimp sequence. So to start off, we're going to click, we can open that chart. We're going to compare the human chimp sequence to each other organism, entering the number of differences in the chart. The tip they gave you is that the human chimpanzee versus horse, note that a minus sign will indicate that an amino acid is missing in that sequence, and when comparing 2 sequences, an amino acid in one sequence and a minus in another would count as a difference. All right, so the minuses are differences as well, so different letters or minus signs, and then you will submit the completed chart and the reflection to your instructor. Okay, and so human versus all these different animals, and we're going to count the number of differences. So then we load up our chart. Okay, and it's going to give us the list. I'm going to make this a little bit wider so we can kind of look here. So we're going to compare this sequence right here to all other sequences on the page. All right, so if you want to use a ruler, if you want to use a piece of paper, that'd probably be helpful. But let's just take, for example, the human chimp sequence versus rhesus monkey. So in my data table, okay, and I'm going to go ahead and show you guys this because some of us are making notes very hard. So when you open up your notes, excuse me, your Evernote, yours is going to look a little different because this is the app. I'm going to get myself a new note. I'm going to title it with Lab 1.05. Okay, and what I'm going to do is make a data table. And I know that in the data table, so right here, I'm going to have the organism that I'm comparing and the number of differences. So it's just going to be kind of a glorified T-chart. All right, so, and I can make it, I don't have to make it quite this big because I know I'm just putting numbers in there. Oh, okay, so for example, we've got the, let's give it a title here. So we're going to say human chimp sequence versus, and then Okay, so my first one, I'll have to modify this. My first one is the rhesus monkey. Whoop, rhesus monkey. Okay. Guys, I'm sorry, I'm kind of doing this on the fly, so I'm going to actually title my table, organism and number of differences. And so my first organism, now here we go, is the rhesus monkey, and let's go ahead and look at the number of differences. So I'm going to use this right here to look at my sequences. So remember, these are missing, so they're the same because they're all missing those proteins. And then I'm going to go letter by letter until I find a difference. K, K, I, F, I. Remember, these are amino acids, M, K, C, S, Q, C, H, T, V, E, K. And so the number of differences between those two sequences is 0. And then I'll go down to the horse. And I'll pull my note down so I can see same number of missing amino acids, G, D, V, E, K, G, K, K, I, F. Oh, here, right here, I see a difference. This has I, this has V, so there's 1. Okay, I see a second difference coming up right next, so there's 2, K, C, 3, Q, C, H, T, V, E, K. So there were 3 differences between the human's cytochrome C and the horse cytochrome C. And you're going to continue doing this all the way down. Reminder, when we have these minuses, this is considered a difference. All right? One of the mistakes people are making is not counting these minus signs as differences. Okay, so once I'm done with the chart, I need to look at the rest of the assignment. Okay, it says, once I get done with all these animals, or all these different species, we're going to summarize your amino acid comparison in paragraph form. To guide your summary, reflect on the following questions. So there's 5 questions that need to be addressed in your paragraph summary. Having to do with what does it mean when the scientists say living organisms share a universal code. This is an objective. You have to be able to answer this question. You're going to have to go back through the module and read it. How does it relate to the hypotheses? How are self-regulating molecules? How might similarities and differences? Okay, on number 4 and number 5, it is very wise that you include your data. So you'll go back to that data table and you'll look and see exactly how close each of these organisms are. Or you could say something, make your claim, your evidence, and then rationale. So how might similarities or differences in genetic codes be used to determine how closely related? Well, you're going to say, since rhesus monkeys have 0 differences, one could say that they're more closely related to humans than the Candida yeast. Okay, and you're going to use that because you're going to say 0 differences where Candida has however many, 19, 20, whatever. All right, so make sure you use data on number 4 and 5. Those questions can be answered right there on that note. So let me pull that up just so you can right underneath your table. You could start your paragraph. You can title it Summary and then start in answering your questions. If you want to do like little bullet points, answer the questions, and then craft your summary paragraph, that's fine too. Just make sure you are in complete sentences. Right? That's as tough as the lab gets. I'm hoping that this has helped you with some of your confusion. If you still need help, just a reminder, once I'm done with that note, I've done my data table with my title and my labels, I've written my summary paragraph, I'm going to retrieve this note link address. And so in my application, I can go to note and copy note link. In the web portion or portal, it's over here on the right and it says share, and then you can find the note link. But remember, that note link is going to give you a URL, and that URL is what you're going to push or post into Connexus. All right, guys, I hope that helps a little bit. If you still have questions, let me know. Otherwise, I'll talk to you later.